See, we just had World Environment Day on 5th of June. What was the theme? Land restoration and stopping desertification and climate resilient system. See, when we talk about climate resilient system or a drought resilient system, engineers' roles are there. You need to come up with technology which are drought resilient. Okay? So now when you look at this slide carefully, what do you see? You have only one earth, and it is the responsibility of each one of you to protect that earth so that your children can be happy. See, when your children are happy, naturally you will also be happy. So, and the best way to respect nature is to hug nature. That's what one of my students, can you see the two fellows hugging a tree? Okay? Okay. Now the goal of anybody, our work is to sustain resources and maintain ensure clean air, water and environment for the children. Okay? When you do that, we say it is a sustainability or sustainable development. That is what we need to do that. Every action of ours, we should ensure that we maintain the clean air and water, etc. Okay? So now if you have to do that, you need to have a responsible decision making. That is where engineers come to the see, when you implement a project, you need to be careful. You should make sure that there is no damage to the environment. You might ask, can we do that? Yes, it is possible. I will show you how to do that. Okay? So now, what is this photograph shows? What does it show? Banyan tree. Aladu Mara. Okay, this is one of the biggest banyan tree in India, which covers about 150 acres of land. But the beauty of that is, look at the way you engineers also design the product, no? The nature is also doing that. When you walk through that, you get the feel of the nature and you will enjoy that, okay? Now let us see the next one. You saw the tree here. Now what is the worth of a tree? See, I am taking a class here. I am sweating. Not because of you people, because of the humidity and the... But if I had taken class under the tree, I wouldn't have lost my water, isn't it? So then how much money you will assign to a tree? See, one is taking a class in an auditorium for 1 hour. What is the charge they pay? We have to pay. That could be the amount you assign to the tree. So in every 1 hour you are paying this much. Then in a year, how much the money you can? But you have other comforts also. It will give fruit, this fruit doesn't give fruit, nor food. Isn't it? Whereas the trees give, and trees give oxygen. This fruit gives what? Carbon dioxide. All of you are breathing, I am sure, no? You are all breathing, no? Is there anyone who is not breathing? I want you to raise your hand, at least for this question. You know you are all breathing, no? Yes. Even that is a doubt. When you breathe, what do you give out?
Carbon dioxide.
Finally, one answer I got. Okay, that is good sign. Now you understood the importance of a tree, no? I'll ask, because all engineering students keep telling me whenever I do counseling that they are stressed. When you are stressed, what do you do? How do you get de-stressed? A mic is required. Mine is a classroom with an interactive session. What do you do when you have to de-stress yourself? out for a walk. The next, best solution, go and hug a tree and shout at tree if you are angry, cry if you are emotionally down. Then you see the beauty of that. You know, same thing happens at home, isn't it? What do you do at home when you are stressed? You go and hug your mother. Don't you do that? Or you don't do that? See, in our generation, no, if mother is shouting, we used to go and hug. Mother used to forget what they are speaking, and we used to have a happy life. So now I have given a technique for de-stressing. You know, I have seen lot of students of engineering, for no reason they undergo stress, because you know they all have become slave of one instrument that is called mobile, and whenever they work they look at that only. Instead of that, had they looked at the tree, probably they would have been happier. Okay, so now you know we have a symposium, that's what Kiran talked about it, called Lake Symposium. This is one of the popular events, you know, a lot of international and national delegates come. We are also giving an opportunity for school and college students to take a part in that. See, when you attend, you know, people like me who have worked for 2.5 decades, we will do the presentation in half an hour. You will know what we have done in a half an hour. Your journey can start from that time. You know, like that there are many experts will be there. And so this is the Aghanashni estuary. It got a Ramsar tag. Why it got a Ramsar tag? Because it was so rich in biodiversity and rich in product. You know, it used to give the fish, sand, salt, worth 1 million rupee per hectare per day, per year. You know, because of that, Ramsar Secretariat has designated that as a Ramsar wetland of international importance. See, when you get this gold medal, you know, what is the advantage of gold medal? You can showcase that you are gold medalist and you are a talented person. Similarly, Ramsar tag has that given to the Aghanashni, has got that. This is in neighboring district, Uttara Kannada district. Okay. So now, see, as an engineer, how you can do the thing? First thing, you can communicate. Take the advantages of the you know, advancement that is happening in information technology. Okay. How do you do that? Create the expert system, decision support system. I'm sure you all know that expert system, decision support system. You can also do the kind of decision, you know, suppose if you have taken some decision, how it has affected the system. You can visualize that only engineers can do. Okay, biologists, we think about it, but engineers can show how much damage. Suppose if the tree is cut, how much oxygen is reduced in the area, you can quantify that. Okay, now this is a website where Lake Symposium details are available. You know, this is the website wgbis.ces.iisc.ac.in. Okay, when you look at this website, there are decision support system, there is a Lake Symposium detail. Now I will show you Lake Symposium, later I will show you decision support system. See, when you click on that, Lake Symposium web page gives the information about symposium, and here students and teachers corner is there. You know, what kind of experiment you have to do, how to do that, and also the videos. We have created some videos related to the subject which is available there. So that if you look at that video, you can also replicate that experiment. Okay? Then we have conducted many events like this, pre-lake conferences in many institutions. All those, the details, you know, the videos, we have put it at the web page. At any point of time, you can also look at that. In case if you have missed now, you should be able to learn from that. Okay? Now, what is our agenda? We are having the Lake Symposium, as Kiran said, during 17th to 20th of October. You are organizing this event. Alvas is organizing the event. Okay. Now, as a part of that event, you know, we allow the school students and the college students to take part. They can do the presentation. You know, about 15 or 20% of the total audience are the school and college students. That means the youth will constitute about 20% of the total audience. And the students, if they have to take part in the event, we have to tell them what kind of experiments to be done, how to do that. That is a part of the orientation program. See, the many institutions organize conferences, many institutions does the workshop, but this conference is different, where the team itself, we go and train the students, so that we want the students to learn the concept and come and do the presentation better than what we have taught them. That is what should happen. So you all will do that? Okay, now I'll ask a question. How many of you will attend the Lake Symposium? I want your hands to be up. Okay, where is Sunita? Okay, good. This is a good sign. There are some, in India, when we vote, only 30% fellows vote. 70% will sleep. And 70% also will vote when people like me start talking. Okay. Now I have started. I will see that all of you do that. Okay. The event is going to be during 17th to 19th October. 20th we are going to the nearby area. Understand how nature works. Okay. Nature functions. Okay. Then of course 20th October I will return to Bangalore. So now the logo of the symposium is this, and this gives you the brand. Okay. Lake Symposium. When you look at this, you will see everything. You will see water, you will see forest, you will see bird. All are interlinked. How it is interlinked, we will explain that. Okay. So now I will teach you. You are all engineers. When you look at this slide, what is this on the left hand side? Okay, you all have learned thermodynamic principle, no? Huh? Is there anyone who doesn't know thermodynamics principles? How many laws are there, thermodynamic principles? I want to answer.
3 laws.
3, very good. Okay, now look at carefully. There is a food chain, isn't it? The food is being manufactured. I am using the term manufactured because you are an engineer. Okay, for a scientist I would have said food is produced. The plants produce the food by taking carbon from the atmosphere, converting it to glucose and store in the plant. That is the food you get. And in addition to that, what does the plant give? Plant gives oxygen. Why oxygen is important?
For breathing.
You all are breathing because of oxygen. Okay. So now the food is eaten by the next level, herbivore, which is eaten by carnivore. When you look at this carefully, there is an energy and material transfer happen from one level to other level. That is the first law of thermodynamics. When there is energy transfer happen from one level to other level, there is a loss of energy. That is called law of entropy. What is that law? Third law of thermodynamics talks about law of entropy. Now you see, by this simple food chain, you have learned the principles of thermodynamics. Will you forget this in your life? Whenever you see plant outside, you will think of thermodynamic principle. And finally, whatever is left in the system gets converted into elemental form by bacteria and fungi. They are called biodegraders or bioengineers. And when you throw mango out as mango rods, after 2 or 3 days, you don't see the trace of mango. That is all consumed by bacteria and fungi. They would have converted organic carbon to inorganic carbon. In the process, you don't have any pollution in the system. So now, when you look at this slide carefully, there is nothing called waste. Where do you see waste?
The litter waste like
very good answer. Very good answer. You will see waste where waste people are there. Who are these waste fellows? Like my friend said, no, are the fellows who litter. If you see a heap of waste in a ward, you can tell them all waste fellows are residing there. Now, look at the second part of the slide. All the plants are competing. Like you all are competing to get highest mark in your class. There is a competition. At the same time, you see there is an optimization. Everyone is synthesizing carbon and sunlight And there is an optimization also. You learn the principle of optimization through that. Now, what else do you want? See, nature is the best teacher. When you do the experiment with nature, you will become a better person. Okay? Now let me take you to my home. My home is Western Ghats. You know, from Kerala to Gujarat is my home. Entire Western Ghat I own. Okay? What is the beauty of that, my home? Western Ghats, it has hundreds of rivers. Because of hundreds of rivers, I have water security for the southern India. Because of water, people grow vegetable, people grow crop, there is a food security. Okay? Now, let us try for a while understand how many varieties of plants and animals are there on the earth. You all said there is only one earth. How many plants and animal varieties are there on the earth? See, when you look at our scriptures, okay, Rigveda talks about 8 to 12 million species on the earth. Okay, that's where you should read our literatures. See, Bible talks about conservation at least 320 times. Similarly, Quran talks of water conservation. You know, if you pollute water, the Quran says there is a war, world war is imminent. So that's where when you visit Dargah, you have to wash your feet and fingers with the clean water. In Dargah, good Dargah, there will be a well. You have to pull water and wash your feet and fingers, then only you can enter the Dargah. So when you do that, they thought that you will practice at home, you will also practice in community so that there is no water pollution. Okay. Similarly, in our literature, see, there are number of species are mentioned. Now, in science, we have said about 1.3 million species are there, and that means million varieties of plants and animals are there. 1.3 million species, we know only 10 or 15% their medicinal properties. You can imagine, had we find out the medicinal properties of all the plants and animals, how rich we would have been. See, that is where talent is required. That is where youngsters are required. That is where youngsters from Alvas are required. They have to do this kind of experiment. Okay? So, now, let me show you which part of the globe, Earth, has the highest number of plants and animal varieties. As we go, you know, you see that highest number occurs at this location. See, y-axis is number of varieties of plants and animals, while the x-axis is the latitudinal gradient. You know, as you move, you will see at 8 to 14 degree latitude, you have the highest number of plants and animals. Highest number of plants and animals are there at 8 to 14 degree latitude, that corresponds to Kerala, Karnataka, Tamil Nadu and Goa part of Western Ghats. How many of you are from Kerala? I can see the face. Make out Keralites. It's not that difficult. How many of you are Keralites? Okay. Why you are hesitant? Kerala will raise the hand without fear. Okay. How many of you are from Karnataka? Okay. That is good. Then how many of you are from Tamil Nadu? Okay. That's good. You understood. We all have got highest varieties of plants and animals in our region. That we have to conserve it. You can consume it. I am not saying that you should not eat. You should eat. At the same time, you should ensure that your next generation also gets these plants and animals. You know, that is where reproduction and all those things you have to keep in mind. Okay? Now, see this. Look at the varieties on our system. How many varieties of brinjals are there? Can you see this badnekai in our system? How many varieties are there? One is good for fragrance, you know, in the South Kerala, we have gulla. Okay? Then we have other varieties. If you make wangi path out of that, you will see the taste of that. Suppose if two different types you make wangi path, you will be surprised to see the taste will be much better. So, similarly, if you can combine those two genes of the two varieties, you get a better product. That is called hybridization. You can think of how to hybridize native varieties to have a better product. Don't think of genetic engineering. Genetic engineering is slightly complex and difficult, whereas hybridization is easier. Okay? And in one of the taluk of Uttara Kannada, we found that 54 varieties of rice, 54 varieties in one thali. See, one variety is good for, making like you use basmati rice in the north, no? Something equivalent to that we have in this district. And other one is for the fragrance. If you combine that, again hybridization, you have a better product. That's what some farmers do. Okay? See, now as an engineering student, now engineer means who? The brain always keep working for an engineer. If brain is not working, that person is unfit to be an engineer. You can have a BE degree, but that BE degree will not be useful. If your brain is equally competent, you become a successful engineer. Your brain can become competitive only when you think differently. That is where engineer has to think differently. How we can use technology and data to do the conservation. See, now you are all engineers. I am sure computer science fellow is there, civil engineer is there, mechanical engineers are there. We launched the satellites way back in 70s. And when we launched the satellite, we also learned how to take a picture of the Earth at regular interval. Every 21 or 30 days interval, we get the picture of the Earth. Now from 1970 onwards, every 21 days, if you are getting picture of the Earth, you can analyze that and say the thing. See, suppose if you have a picture of yourself last year and this year, what is the use of that picture? You can find the difference. You can find the changes. Now, if you have a picture of the Earth in 1970s, 80s, 90s and 2000, I can see what changes are happening. See, that is what we have done. The changes over a period of time using the remote sensing data. Now, to analyze that, because they are grey level, you need to code that. Earlier to classify one district, we used to take about a year. Today, because of artificial intelligence and machine learning, we have become faster. Our Tulika will tell you how to take advantage of artificial intelligence and machine learning and become better than what you are today. See, your natural brain is the best. The artificial intelligence means you can make it still better. So that is where, look at the thing. I am talking about nature conservation. I am also talking about artificial intelligence, machine learning. That can happen only with my subject. Whereas other fellows, mechanical engineer, he will only think about the bolt, nuts, etc. Computer fellow will think about only bytes and bytes, okay? So, whereas we talk about all the subjects, engineering, biology, science, etc. Okay? Then, we also calculated what is the carbon that is stored in the plant. See, how do you measure the carbon in a grassland? You will put a plot of 1 meter by 1 meter, cut all the grasses and then measure the weight. And you will say 50% of that is a carbon. Now, if the same thing you have to calculate to find out how much carbon is stored by trees in the forest, what do? See, we said that because of the photosynthesis, there is a carbon sequestration happening. And because of that, oxygen is produced. You want to calculate how much oxygen is produced by the tree. Then only you can say that what is the money for that oxygen. To do that, you have to find out what is the carbon that is stored in the plant. Once you know how much carbon is stored, the converse of that is how much is oxygen produced. Okay? CO2 breaks into O2. That's what happens. Now, see, how much carbon is stored in the plant. How do you assess? One way is cut the tree, weigh it, and you say 50% of that is carbon. But cutting a tree is a disaster. You should not do that. What do? We have an indirect method. What is the indirect method? We will find out the height of the tree and the girth of a tree. With that, we find out what is the volume, and from that we find out the biomass. Once we have the biomass, we know that what is the carbon that is stored, 50% of that. Now, if I want to find out the weight of yours, how do I assess? I put you on a weighing machine and find out the weight. But if I don't have weighing machine, what I will do? I will ask you to stand up, find out what is your height and what is your girth, the circumference. Then with that I will say what is your likely weight. You know that For this height of 125 centimeter, you should be around 52 kgs. That relationship you know, no? That means for every height and girth, we know what is the biomass. And that is what we do for carbon estimation in tree. That's how we did for all the trees in Western Ghats. See, that's the beauty of science. You do small experiment and then take it to the larger area. Okay, so that's what we have done. See, this is above ground biomass, this is below ground biomass, and this is soil carbon. Okay, this gives the total carbon. Now, when you look at this total carbon that is stored in the trees, it amounts to 1.23 million gigagram. And every day carbon storage happens. How much happens per year? It's about 37,000 gigagram of carbon gets stored in the trees every year. That is equivalent to the emission that happens in Mangalore, the TVRAMACHANDra, Kochi, Bangalore. That means nature knows how to balance it. It's taking the carbon, and that's how it is maintaining the balance. Now, what is the money value? See, Dakshin Kannada people know, only they talk about money. When I come in the bus stand, no, first question they ask me is, how much money you earn per month? They don't ask how good you are or whether your health is alright. First question is, what is the money you earn? Okay. Suppose if somebody comes with a son or a daughter, first thing they ask is how much money son earns or a daughter earns. So that is where money, that is very important. Now what is the money value to that tree? The work that is doing, it amounts to 100 billion rupees or a $1.4 billion. Now what is the use of this number? See, when I go abroad, when I represent a country, I'll tell the abroad fellows, see, all the trees in my country, in Western Ghats, is taking carbon from what you are producing because of your industrialization, which is stored in my trees. It is amounts to 1.4 billion rupees. You should give 1.4 billion rupees because I am maintaining the forest and doing the job of carbon sequestration. But I won't reveal them because of the trees I am getting oxygen. I will just tell them that because of you, whatever the carbon you emitted, I am taking it. Okay, that's what is happening. Okay, is it clear to all of you? So now if this continues, the students in computer science would have done the modeling, isn't it? Agent-based modeling, etc. Now we use the same thing to say what is the likely situation. We found that there is a further reduction in the forest cover in the Western Ghats. Now if there is a reduction in the forest cover, what is going to happen to the climate? Let us see. See, the Kerala will become warmer. That means temperature in Kerala will go up. Then Karnataka, the temperature will go up. You all saw now recently how temperature increased in the region. Okay? And then second thing is rainy days. The rain which is coming, Kerala receives highest amount of rainfall, will start getting lower rainfall. That means they cannot practice the cultivation of some of the crops like areca, sugarcane, etc. Then the most worrying thing is number of rainy days. Rain is spread across the 4 to 8 months. Instead of that, you get a rain in a short duration. When you get that large quantity of rain in short duration, that is called high intensity rainfall. When you have high intensity rainfall, what will happen? Flooding will happen. With the flooding, landslide, mudslides will happen. See, now you see that how the things can be changed. If you have maintained the vegetation cover, you wouldn't have changed the climate. So that's what is to this. See, the 4 decade or a 3 decade, there has been about 10 to 15% forest cover in the evergreen forest cover loss in the Western Ghats. That's where we are paying the price. And if the temperature increases, I said Kerala become warmer by 1.5 degrees Celsius, what will happen? You will have more and more diseases. Chikungunya, dengue, these are all vector-borne diseases. That means the vectors like the mosquitoes will increase. All those vectors, once the vectors are more, naturally the diseases will be there. Now you can see how engineers can do all this analysis. See, he or she can talk about the medicine, he or she can talk about what is likely to happen to the society, how many diseases will come, etc. Can anyone else do this? See, that is where, when you do the experiment, if you take up the innovative experiment, you can think differently. That should happen, okay? So now, see, when I was of your age, I used to have water, but now when I go there, no, I don't see the water. What is the reason? This again engineer applications, okay? Huh? Because of, you are sure? You have which background? Huh? BSc, okay. Okay, now let us try to understand this. For that, you know, it is like this. How the vegetation cover in the region decides the ecosystem functions. You know, what are the ecosystem functions? The ecosystem does the job of hydrological cycle, nutrient cycle. Just now I talked about carbon. Hydrological cycle, water cycle. Can you tell me, any one of you, what is water cycle? The person who has not opened the mouth so far. No, I see many people opening the mouth in different way. When I ask questions, they open. Some people once in a while they are opening the mouth. Huh? Okay. Who will tell me this? The water cycle, who will tell me? You will tell? You are showing me water bottle which is empty. That is like today's slide. What is the point in me having water bottle without water? Huh? Okay. Huh? Yes. Very good. So Kiran is your Kiran now. See, not only he is culturally rich, he is also subject-wise he is the confident. Okay. I will tell you one instance where I was invited, you know, Andhra Pradesh. I was supposed to take a class for faculty from engineering colleges. There was 60 faculty were there in that class. I asked what is water cycle. Like Kiran, one fellow got up. But unlike Kiran, he said, sir, see outside. I saw outside, thinking that it is raining. Then he told me, sir, you see outside, people are carrying water pot on bicycle. When you carry a water on cycle, it is called water cycle. What do you do with that kind of teacher? Huh? 45 or 60 of them were there. I just walked out of the hall. I said, I don't want to continue with this group. Then of course, people came and said, just because of one apple, bad apple, why did you walk out? You can take the class for us. I said, no, I will come back after 12 days. You are all here, because 15-day program. I will come back and take a class again for the people without that bad apple, I said. See, that is the problem. If you do not know the subject at young age, how disastrous you could be when you become a professional. Tomorrow you might become a faculty. Tomorrow you might become an engineer in an industry. When you become an engineer in an industry, when a child at home asks what is water cycle, if you tell when you carry water bicycle become water cycle, then what is your child will be? It become one more water cycle fellow only. So do you want to produce the children like that? See, then only you should understand the concept clear. That is where, you know, when you attend classes, when you are attending, the things will help for you. So now I will take you to the example what I was telling you. Let us go to Sharavati River Basin. See, it originates at Ambutirtha, traverses 128 kilometers, joins the ocean. See, these are all the projects. You can think about doing the similar thing in your area. That's where I am sharing some of the examples. You know, now dam was constructed in this region. Okay? When the dam was constructed, you know, there were 179 villages were there. They were removed. They were put in a dryer bed. You know, when they are put in dryer bed, because these farmers were doing wet cultivation, they knew how to do the cultivation when there is water, but they didn't know how to lead a life without water. What they did is, they waited for the reservoir to be completed. Then after that they came back to this region, they converted forest into agricultural land, and today agricultural land has become a barren area. Okay? Now one mistake of engineer, see how it has complicated life. Now let us see how it has affected the rainfall. See, to see the rainfall, you get a rainfall data from the last 122 years. You just download the data, analyze the rainfall for the period 1901 to 1964, then 1965 to today. You will be surprised to see 1901 to 1964, entire region was receiving the rainfall of 3000 to 4500. That means entire catchment was receiving the rainfall. I am sure as an engineer you know what is catchment, what is watershed. Jala Nayana Pradesha. That means when it rains, the water gets drained to that water body in that region. Now the entire region was receiving 3000 to 4500 millimeter. Now if you see that today, 1965 to today, this portion gets the rainfall of 3000 to 4500, while this region gets the rainfall of 1700 to 1900 millimeter. You just overlay the vegetation cover. Wherever vegetations are there, wherever forest of native species are there, you have rainfall of 3000 to 4500. Wherever forest is declared, you see the rainfall has come down to 1700 to 1900 millimeter. Don't you see the thing? To analyze this data of 100 years, engineers are required. See, that is where the engineering brain are required. You can automate the whole process through the machine learning algorithm also. Okay? Now, we see our interest is not just the rainfall. How long the water will be there in the region? For that, what did we do? We put a sensors in the underlying catchment. You know, see if the catchment is with the forest cover, we put a sensor there. Similarly, if the catchment is dominated by urban area, we put one sensor there. Like that, about 28 locations we put the sensors to find out when it rains, how much water goes to the underlying. See, you all design sensors, no computer science student. When you design sensors, you know, now IT enabled thing you know is gaining the prominence. When you put that, it detects how much water goes to the underground. When you quantify that, you know, when it rains, if the catchment is dominated by native species forest means it will be diverse. When it is diverse, roots are diverse. When roots are diverse, you know, soil microorganisms are diverse. When soil microorganisms are diverse, the soil become porous or permeable. When soil become porous or permeable, it allows the water to percolate. That means when you put a water on the soil, water goes to the underlying. If you put water on this, what will happen? Water will be there on the land itself, because this is non-porous surface. Now when you have porous surface, when the water flows, what is the advantage? Water fills in this region, you know, this is groundwater zone, then it fills this zone, water zone. Then after that only you will see water flowing in the stream or a river. So now what is the advantage of water filling in this the underlying layer? You have rain for 4 months, you have water in the water body for 4 months because of the rain. Then which happens in all water bodies, okay? Then after monsoon, the water that is stored here will move to the water body for the next 4 months. Then the water stored in the groundwater zone moves to the water body for during the summer. That is called the base flow. You know, in hydrology chapter you would have understood the base flow. Base flow is the flow during the summer. Now see, the water body will have water provided the catchment has the native species forest. If the catchment is like urban building, you will not have the groundwater recharge. That's why the people face the water crisis. See, as an engineer, you can tell why the water crisis happens. Okay? Now, see, we graded the streams as A, B, C, D, the water body as A, B, C, D. The A are the one which have the water body, water throughout the year. B are the one 8 months, C are the one 6 months, and D are the one 4 months. Now let us see where A, B, C, D kind of water bodies are there. See, A type of water body is there. That means this water body has 12 months of water provided the catchment has the vegetation cover of more than 60%. That means in Jalanayana Pradesha, if the forest cover is more than 60%, your water body will have water for 12 months. If we convert that catchment into monoculture plantation of rubber or acacia, then what will happen? You will have water for 8 to 6 months. Okay, that is what is shown by B and C. When we declare the catchment with the forest cover less than the 40-30%, you have water only during the monsoon. Now you will ask the question. Nangain Prayojana, because South Kerala people know. So now I will show you how that Nangain Prayojana I can answer. See, in this region, people get water for 12 months, here 8 months, here 6 months, here 4 months. This analysis can be done only by the engineers, because spatial analysis, remote sensing data, all those things can be done by the engineers if they are trained properly, if the students have attended the classes properly. Okay, now see, here I said 12 months of water, here 8 months, yellow is 6 months, 4 months. Now you just overlay the vegetation cover. Wherever the vegetation of native species is there, you have 12 months water. Wherever vegetation is fragmented, depending on scale of fragmentation, you have 8 to 6 months of water. Wherever the forest cover is less than the 30%, you have water only during the 4 months. Now let us see what is the revenue people get. See, farmers in this region, because water is available throughout the year, they can grow multiple crop or a cash crop. Whereas here water is available for 4 to 6 months, they can grow only one crop. See, when you grow only one crop, your earning is 32,000 rupees per acre per year. Whereas in this region, since farmers are growing the multiple crop, because water is there throughout the year, their earning is 1,54,000 rupees per acre per year. Now another interesting thing we found out was, in this region, what is the demand of water in various sectors. See, we calculated the demand. This is a demand curve. You see that demand is much lower than the availability of water in this region when there is a water surplus is there. In this region, because of degradation of forest, you see demand for water is there, but not sufficient water is there. Okay. Now, how much water get infiltrated during the monsoon when forests are there? We did a quantification. It shows about 60 to 65% of water gets infiltrated. Whereas in this region, about 28 to 30% of water gets infiltrated. This is the work our Dr. Vinay did for his doctoral work. Is it not a good work? He is a civil engineer. But he thought about nature. See, that's how you can also do the work. That do your project, your final year project, you can design like this. For that, this kind of forum will help you how to think differently. Then another interesting thing, because there are students from science background. You see the pollinators in this region are abundant and diverse. Whereas pollinators are fewer in number. When pollinators are abundant and diverse, what will happen? Pollination become efficient. When pollination become efficient, what will happen? You will have higher yield in the crop. Because of the higher yield, see, here farmers get, for example, paddy, they get about 14 to 18 quintal per acre. Whereas in this region, since there are no pollinators, people have degraded the ecosystem, there are no pollinators, the yield of the crop is 6 to 8 quintal per acre. Now 6 to 8 quintal per acre is better or 14 to 18 is better? Naturally 14 to 18 is better. For 14 to 18 is better. Why? Because the water is available. Because of the water, there are pollinators are higher in number. Why pollinators are higher in number? Water is more because the forests are there. Native species forests are there. Okay? See, now let us try to understand this. See, all these engineering students can do. See, now we map the other animals like tiger, sloth bear, etc. They all are there where water is 12 months. Where water is not there, only two-legged animal is there. Who is that two-legged animal? All of you are two-legged animal. Okay? See, we also found two species of the fish which has a medicinal value. What is the medicinal value? It can be used for curing the asthma or bronchitis. I thought that because it can be cured, has a medicinal property, I should become rich. I took the fish to the Bangalore. I cultured in my lab. When I cultured, none of them had any medicinal properties like Bangaloreans. So then, what is the message here? If you want medicine, you have to have the fish in their home, in their habitat. When you have them in their habitat, for the fish to survive, you should have water 12 months. For water to be there in that water body for 12 months, you should have forest in the region. See now how the linkage between the forest and fish has come. Okay? Then, you all know frogs, no? Frogs are known as amphibians. Why do we call frogs as amphibians? One at a time. Who wants to answer? I think our HOD will give the mic. Yes, Prasanna.
It can be in both water and land.
I was also in the swimming pool in the morning. Just now I got up and came. Can you call me amphibian?
It can breathe. It has
It has.
the ability to
I have the ability to stay in the water.
breathe.
See, that's where whenever my student asks question, no, I get into water body and stay there for up to here. Okay. I will answer the question. See, that is where, yes, that is a good sign. Yes.
It can control its body temperature when it is underwater and on land.
Okay, I have still not monitored, man. I will take it up as the next experiment. Okay? You understand? See, I always learn my subject in a simple way. You know, frog has two stages in its life cycle. First stage is a juvenile stage, like you all were child, no? Like that juvenile stage. Juvenile stage is a tadpole stage. Tadpole means fish-like structure. That is in the water. Next is an adult stage, which is on the land. Because of two stages in the life cycle requiring two different the habitat, frogs are known as amphibian dual life. Your answer is correct or my answer is correct? Huh? You understood how to learn the subject, okay? So now this kind of experiment you can do. See, what we saw is, we come across one frog in a dry area. We were surprised to see. We wanted to see, okay, how this frog will survive. We started observing that. When we observed, we found that this frog, after fertilization of the egg, we saw the younger ones were coming out. Why? Because there was no water. The frog has learned how to modify his, reproduction process. It bypassed the tadpole stage. So that younger ones came out. Then I was curious. I told my student, get that fertilized egg to Bangalore, let me see. When he got to Bangalore, on the 14th day, I saw this is the younger ones were coming out. That means the frogs have learned how to get adapted to a situation when there is a water scarcity. What do you say? Without water, can you survive? Because you are closing your eyes, meditating to tell me that what a situation we are reaching. Correct, no? Huh? Can't survive, no? That is correct. Okay, please be seated. No? So now, you understood the thing. This is called adaptation. When we talk about changes in climate, mitigation, that means reducing, and adaptation, getting adjusted to a situation is the thing. See, one other thing, the process happens in the biological organism is the reproduction. Because of reproduction, there is a renewability. You see, younger plants come. Had the younger plants have not come, then you wouldn't have sustainability in the system. So that is where you need to have the younger plants. Similarly, in the humans, similarly in the animals, etc. Okay? Now, we thought, okay, these are very important. Science should identify the region which are very important. See, that is where engineering students are required. See, what we did is, we developed the decision support system. We looked at all the variables, biogeoclimatic variable. We put it together and then identified the region which are ecologically sensitive. We divided the region into 5 minutes by 5 minutes grid. In each grid, what is the forest cover, what kind of animals are there, which one is endemic to the region, what is the rainfall, all those things we put together. We came to know which are highly important. Like in your score, you have CGPA. How do you get CGPA? You have the score for each subject. When you add that, you have a consolidated score. Similarly, we found out CGPA because all the marks what we have got, we converted into weights. See, if it is 80% forest cover, 10. Like that, if it is 60 to 80% forest cover, it is 8. Like that, we converted into the marks. Okay, then each grid got the total marks. Then we divided into 4 groups. The CGPA more than 9.9 is considered as a distinction. No? Like that we did. And we said which are ecologically sensitive. See, now these are ecologically sensitive region. Green, dark green is ESR2, light green is 3 and blue is 4. Now sitting at home, you should be able to find out which is ecologically sensitive. This is a job done by engineers. You can do that. For that you should know the database. database management and all the algorithm behind that. That can happen. See, this is a decision support system which is available online. See, who all are the others? All these people are worked. There are engineers, there are computer fellows, okay. Now when you access this, okay, you will see the, see, I will take you to that portal. From Mudbitra it is going to Bangalore, no? That is taking time. Okay, I will, see, this is the portal where the decision support system on the server is there, and also we have app also which is there in the Google Play Store. It is called Sayadri. See, here you click on the spatial decision support system, okay? You got it. Now if you want to find any region, for example state, Karnataka, okay, district. You can choose the district. I will choose school, okay. See, here I come to know which village is ecologically sensitive and why it is, okay. Then if I want taluk, I can choose the taluk also, okay. Now in a taluk I will come to know the exam. Then I can also say that which village, okay. If I click on that, I will come to know. Like here CGP, you know, which subject you got high, which subject low. Like that, what is the forest cover, what kind of species are there, all those things. And all the data what we use, we have put it online. Okay. Don't you think the role of engineering students in this? See, this can be done by you people. This is called the decision support system. You know, decision support system, planning support system, expert system. These are all the hot subject in today's this one. If you want to be successful in the interview, if you read up this and you want to talk to anybody, they will be excited. They will say, okay, you are most welcome to join the thing. Because you need to develop the apps which will help the society and in the decision making. Okay. Now, okay, I will minimize this. Okay. Okay. Now, I will take you to the field. I will also teach you what is sustainability. Okay. Now you see this lady. She is about 65 plus years. You know, 65, very young lady, no? No? What's all your age? You are all,
20.
in the beginning of first 30 years, no? I am telling you that I am looking at a lady who is in the fag end of the last 4 decades, okay? So she is saying, why she is saying, see she goes under the water and spends 4 to 5 minutes and collect a bucket full of bivalves. When she gets the bivalves, which is called chippu, you know, in this region you know that. When she sells that bivalves in the market, she gets 350 rupees a day. She won't go and collect the second bucket. You know, she tells me that tomorrow I will go. Today I got 350 rupees which is sufficient to run my family. For tomorrow I will go and collect tomorrow. So that is what is sustainability. If you do not do the over harvesting, you know, that is the sustainability. You know, some people know you can wear about 4 or 5 t-shirts in a year. Some people know they will buy 100 t-shirts. See the stupidity in their brain versus just because their father can afford, no, they sit in home, they will buy it through the online. See, whereas this lady says, no, for me 2 t-shirts is enough. One for today, one for tomorrow. When I wash this, I need one for the tomorrow. I don't require one. That is called sustainability. Not over harvesting. Okay? Okay. Now, you see that how the fate of the region is decided. You all know the Easter Island, no? Easter Island. You know, people went on expanding island, thinking that land is available. But one day they have to close the island because there was no water available. See, that means availability of water decides the fate of the island. Okay? The next example is the Vijayanagara kingdom. Last leg of Vijayanagara kingdom failed because of the water crisis. See, now you would have seen my quote here. See, initially I started with science. When I came to this lady, when I interacted with the society, I learned something. Some principles for conservation. Now, when I look at the scriptures and literatures, that is the cultural aspect, I learn. That means all these are required for conservation, okay? So then, look at the practice. Our elders protected a patch of forest thinking that with the forest is there, their wells will have water. See, you see the Lord Shiva, his head is always covered with the hair. As long as hair is there, he retains the water. Have you seen bald Shiva? No. See, when you have hair, your head is cool. See, that's why my student also maintains that, like Lord Shiva. Okay? So that he is sitting there. Always he is cool and calm. Anytime you provoke him, he will smile and talk to you. If he is bald-headed, what would have happened? Monday busy would have happened. Monday busy in this region. Okay. So now see, unfortunately people are removing this forest and converting into idle land. See, that is idle people will do that. So that is the degradation is happening. We need to tell them that forests have a role to play. Bana has a role to play. Plants are there. If Bana is removed and then if they convert it to temple, what will happen? All those Daityas will create a problem as it happened in Kantara. Okay?
So at the beginning, sir, you told about thermodynamics law and biodegraders, and then you came to Sharavati Basin and you told that 179 villagers have been removed, and those villagers were removed because of the dam construction which took place there, and the dam construction affected rainfall.
Dam construction removed the forest cover which affected the rainfall. Okay? If you say just rainfall, then somebody will come and say, sir, reservoir is there, water is falling. Huh? Okay.
And then because of degradation of forest, there is more water demand in some of those areas. And then you told about frogs, they're amphibians, like they have two stages, juvenile and adult phase. And then you spoke about decision support systems and sustainability.
Okay. Now what I will do is, are you choosing somebody from that side?
Hello, sir.
Yeah.
First, you started with this is the Conference of Lake 2024, 14th Biennial Symposium, and you started with the value of one tree and the shade providing, and the banyan trees which providing the shade which covers 150 hectare of land. Then you gave a link where we can search all the details of the lakes or the research articles provided there. Then you gave about the land cover changes and rainfall occurring in the region, and the groundwater recharge and base flow. When the rain comes in the monsoon, how it enters the groundwater, and in the summer season how the groundwater flows to the basin. And you talked about water availability, demand and livelihood, and fauna diversity, vegetation and water linkages, how the frogs will adapt the environmental changes and survive its life. And you talked about medicinal fishes, where you took the fishes to the home and it did not have the medicinal effect, but it had a great impact in that region. And you talked about how conservation, how we should conserve, how women conserve, teaching us sustainability and not over harvesting. That's it.
Okay, thank you. So now what I will do is, I will take for another 5 minutes because we have a tight schedule. I will not get into the details. Recently Bangalore faced a severe water crisis. You would have seen that, See why it faced the water crisis? Because of we get 55% water from Kaveri. There was no water in Kaveri, and 45% groundwater. Groundwater, the table was depleting in Bangalore. Because of over exploitation, there is no groundwater recharge was not happening. You know, when you look at Kaveri, this is the work recently our Vinay did. See, 1965, 33% forest cover in Kaveri basin. Today it is 18%. See, everyone wants to be young. 18 to 20 is a young age. But cutting the forest and making it 18% is not correct. Okay? So unfortunately, when it happens, you will not have rain. When you don't have rain, naturally water shortage will happen. Then the second thing is, if you look at Bangalore, lakes are interconnected. We have lost the interconnectivity. There are, every fellow has encroached the Rajakalve. Rajakalve is the drain connecting from one lake to the other lake. So, you know that if you go to Rajarajeshwari Nagar, where our Vinay resides, next to his house, one film actor is there. He was sitting on the Rajakalve itself. His house is on the Rajakalve. So, fortunately, recently, he was shifted from there and put in Parappanagraha. So, his name is Sarathi. See, people who abuse Mother Nature will abuse everybody. See, people who encroach Rajakalve, that means they are abusing Mother Nature. Naturally, they would abuse the mother, they will abuse the wife. That is what happened. Now, he is under the shades of Parappanagraha. Leave him. So, that means there is something will happen when people do bad thing. Okay? So, now, let us see why Bangalore is facing the crisis. See, it is because of this. See, 68% green cover in 73. It has become 82% red. Red means building. Here, buildings were less than 8% in 73. None of you were born. At that time, Bangalore was a garden city. Okay? Then 92, when your parents appeared in the scene, to give them a job, industries came up in Pune and Koramangala, changes happened. Okay? Then 2000, IT-BT fellows started. So that's where outskirts you see changes happening. Then further intensification, then 2009, and see the kind of urbanization that has happened. From 8% red to the 86%. If this trend continues, we have reached a situation with oxygen deficiency and the water scarcity. There is a 1078% increase in periods of rain, 88% loss in vegetation, and 79% loss in water bodies. See, we have, you all eat rice and that kind of item, no? In Bangalore, people swallow lake, trees like that. Each tree they will swallow, okay? Because of them, Bangalore is dying, okay? So now, see, every year how things have changed. This analysis is done by Dr. Vinay, okay? And we crossed the carrying capacity of the region, okay? So now, we also looked at how many trees are there. Our studies show for every 7%, one tree is there. You might ask, how many trees you require? You all said you are breathing, no? How many trees you require to meet your the oxygen demand? Huh? I have written there, okay? 78 trees. That is because when you breathe, do you know how much carbon dioxide you give out? See, you all give out 980 grams per person per day. Okay? People like me, 540 grams. People who are aged, okay? The senior people. Now, 1 hectare of tree sequester the carbon to an extent of 7 to 8 tons per year. By that ratio, for every human being, there is a 7 to 8 trees are required. See, now we have the oxygen deficiency in Bangalore. Okay? And the lakes, you see, the lakes are, we lost 79% of lake. See, all fellows who swallowed the lake are all sitting in this region. Where red is there, the lakes we have lost, okay? So now, we thought that we need to have the decision support system, okay? For the decision maker. We developed the decision support system, which is called Bangalore Urban Information System. It gives you an idea about how urbanization is happening, where is the tree cover in the city, all those things we analyzed, okay? So, now, see, you can also come from here, BOIS, okay? So it gives you the picture. Similarly, we developed the Bangalore Lake Information System, which gives the lake-wise the information. I think there is a net problem. You can check this thing, Bangalore Urban Information System and Bangalore the Lake Information System. Both are available online, okay? And also the apps are also we developed. It is available in the Google Play Store, okay? Now with this, because of the time constraint, I will not wait for that to respond. This gives the Bangalore Lake Information System, okay? Now.
No, I want louder answers and I want an interactive session and I want you to interact with me every slide. Whatever questions you have, you can ask me. And I want louder. No need to pass the mic. I know you are all loud. When we are in the cafeteria, I am in the college only. So I know how much loud we can be. Okay? Okay. So, good morning all.
Good morning.
Thank you. So, today I will try to show you how to monitor and map your neighborhood. So, you have seen how sir has already explained different aspects of the biodiversity monitoring and mapping. So, I will try to give you a glimpse and how you can also do it. Okay, are you ready?
Yes.
Are you ready?
Yes.
Okay. So, this is we require for field or GPS. That is, you all know, already available in our phone also, right? In our mobile phone it is there. So, to use this in the field to collect the data, we can use some applications, and to map that data and analyze the data, we use some applications when we come back to our lab. Okay, so I will give you some name of some apps and then their applications also, so that you can also do it. Okay, done? Okay, now?
Yes.
All of you?
Yes.
Okay, so first app is GPS map camera. You can go to Play Store, you can install it, and this works simple like a camera, but the main thing is it will give you the location, time and date also of that particular region where you have taken the point. Okay? So field data collection is easier. One thing you have to keep in mind that you have to have the net connection, then only it will work. Okay? First one is GPS map camera. First one is louder.
GPS map camera.
More louder. I know you can be more louder. Okay, thank you. Then next one is Google Earth in Play Store in your mobile. You can go to Play Store and search for Google Earth. You can install it. It works simple like Google Maps. When we click on the round thing, it will track back us to our current location, right? Same like that. You can take your wherever you are in your field. You have collected the point. You can save it and you can use it when you come back. Okay. Now next one, we have taken the field points. Now we have come back. Now we have to analyze what data we have collected, right? So for that, first software I will tell you, that is Google Earth Pro. How many of you know about Google Earth Pro? I want to see the hands. Yes, 2. Only 2 people. Can I see long hands of who knows the thing? I know she knows. Sir knows. Okay. Okay. So the Google Earth Pro, this is. I know Pro sounds like it is a paid app, but it is not. It is free. Okay? You have to go to Google, search for Google Earth Pro. You will get that Earth version link. If you go to Earth version, you can install it. Simple. Just you have to click on OK, it will be installed. Okay? So now the points that you have collected in your field, that you can put in the Google Earth Pro and you can map it. Okay? In Google Earth Pro, we can make points, lines and polygons. Can anyone of you tell me what is point, line, polygon? What is point? Huh? What is point? What is a point?
A circle point.
Circle?
Point.
What is a point? What will I represent by a point? Who is answering? spot. Yes, correct. Anything else? What is the point? Anyone else from the back? Come on. You all know this.
Point is nothing, it's a one-dimensional object. Most of the times when you are representing any two-dimensional object, we can go for
Correct, sir.
one-dimensional
Correct.
point. Yes.
So, in terms of spatial mapping, point is a like current location. If I take my current location and send you, that is a point location. Right? Right, na? Yes. Okay. If I send you Vanessa's location, that will be a point. Polygon location or point location? Point location, na? No confusion? No. Okay. So what is a line? Roads, correct. Anything else? Huh? Rivers, okay. Anything else? What can be lines in spatial mapping aspect? Come on. Roads, rivers, yes?
We join 2 points.
We join 2 points. That is a geometry answer. I want to give you in terms of whatever you can see. Okay, how can you represent a line? Highway, okay, that is a road only. Then, tracks, railway tracks, correct. Anything else? Canals? Sir? Rivers, yes. So now you know what is point, what is line. Okay? Okay, finally, what is polygon? Lakes, very good. Anything else? Forest. And your campus?
Yes.
Your campus? Okay, that hill I can see outside? That hillock, acacia field hillock. That will be point, line or polygon? Polygon. Okay, each acacia tree will be points. The road that will take us to that hill, that will be
Line.
So you all know now what is point, line and polygon, right? You have gone to field, you have taken the data there. Now we have come back. Can you see here? Can you see here? I have taken some points here, suggesting demarcating the departments in my campus. Okay. So and this suggests the roads that connects one department to another, and this is the entire campus. Okay, so can you all do the same thing for me?
Yes.
So this is your quest. Okay, you all have to mark your different departments in your campus. You can mark the roads joining from one department to the other, and you can mark your campus. All you can do, yes or no?
Yes.
I can't hear you.
Yes.
I cannot hear you.
Yes.
Okay. So, to take the point, there is an option called Add Place Mark in the Google and Pro software. Okay, it will be there in top toolbar. What is the option? Add Place Mark. Okay, so to mark a particular point, the appropriate symbol is this white and black round thing. This white and black round thing is the appropriate symbol to mark a particular location. Okay? So don't get confused between many symbols. Then next one is the line. To add a line, there is an option called Add Path. Using this, you can mark the lines from one department to another, or a river or a railway track that you want to mark. Okay? And in this software, this also helps you to identify, note down the measurement also, the length. Okay? You can go to the measurement section, and there the length will be there. Then there are multiple units of measurements are there, you can take it. Okay. Then finally is the polygon. That is option called Add Polygon. Okay. Polygon also you can calculate the area and the perimeter. Okay. Easy? Not complicated, right? Okay. So now we will move on to the next, or is there any confusion till this point? Point, line, polygon is clear, na? This is basics of spatial mapping. Okay? Now, to mark any polygon or to mark any particular area, there are some conventional colors. Okay? NIISC in Hyderabad, how many of you know? Yes. So the ISRO everyone knows, right? The maps from ISRO that we see, which gives idea of where the forest is, where the water body is. How many of you have seen? It comes in newspapers, in news channels also. Sometimes it comes when we scroll the Instagram and Facebook. There also it comes, some of the maps. Have you seen? You have seen, right? Some maps show that tree cover is this much, tree cover has gone down or tree cover has gone up, right? So to identify particular land features, okay, that okay, this area is forest, this area is water, this area is building. To identify that, we have some particular colors that NRSC in India they have already given, so that we don't get confused. Okay, you can go to NRSC website, you can get the colors. Here I have given the colors. You can see it in NRSC website also. Which website? NRSC. Okay. Now this Google Earth Pro application or software, you can say, this also works as a time machine. How many of you know time machine? Time machine. Doraemon also has it. Come on. What is time machine? We can time travel, correct? You have seen Doraemon, right? So here also we can go back to the past. See, you can see this clock-like thing, na? You can see? दिख रहा है? वो ऊपर लाल में जो clock-like thing. Okay? When you click on it, this bar will come where this from 1970s to current image will be there. If you slide that bar, you can go back in time and see what was the situation before and what is the situation now. Okay? See here, this is the image of Haryana that is in this campus only, right? Where the lake is there, bird park and butterfly park. Can you see what was the condition in 2015 and what is the condition in 2024? All of you can see, na? This will help you understand the changes. You can do the change analysis from this cool app. Cool, right? Okay. So, going to the next, you can also save the points in .kml format. Okay? Please do not forget this .kml format. Okay? Which format? .kml. Okay. Next, you can also save the map as an image to use it in your project. Okay? So that you don't have to search in Google and ask for, okay, give me a map of Alvas. You can create your own maps from now onwards. Okay, na? From now onwards, you will create your own maps. Don't ask that, what it is called, Copilot to make a map for you. You all know Copilot, I know. Okay. Then coming on to big data. Big data is, we can say from the name itself, we can see big data. There is a lot of data that all satellites and all the things they are collecting. That data we can take and analyze it for monitoring our ecosystem. Okay? There are multiple resolutions, or we can say that some are 100-meter data, some are 30-meter data. Then some satellites give us data in each day, some give us in each month. So there are multiple data there, thousands of petabytes of data are there. So that data we can analyze using your artificial intelligence and machine learning. I'll show you one cloud-based, we can say, application that you can use to analyze those big data. Okay? Interested? How many of you are interested? I want to see hands. Long hands. Okay. So to analyze the big data, we can use Google Earth Engine. For using Google Earth Engine, you don't need to download anything. It's like your Facebook. You can go to your browser, you can just search Google Earth Engine, you just have to sign in, you will be inside. Everything is in cloud. You don't need to download anything, you don't need to upload anything, everything will be there in cloud. Okay? And it is free also. No need to pay. Easy? Next is what we can do in Google Earth Engine. First is, there are multiple satellite imageries that we can download. Okay? Then next one is the corrections that were required that we do when we upload our photo in Instagram. है ना? we edit the photos. If it is tilted, we make it Sida. If it is, the color is not right, we correct the colors. Okay, that corrections also for satellite imageries, not our imageries. For satellite imageries, we can do it in Google Earth Engine. Okay, then also visualization and analysis also we can do, and accuracy assessment, calculation of the statistics, all the things we can do in this cloud-based application. Okay, which one? Which app we can do? Google Earth Engine. Okay, don't get it confused with Google Earth Pro. This is Google Earth Engine. Then as I said, there is like huge database. More than 10 petabyte of data is there, publicly available. You don't need to pay, you don't need to do anything, you don't even need to download it in your system. You will say my system do not have a space. Everything will be processing will be going on in cloud. So even if you have a basic laptop or anything, even if you are using your friend's laptop, you just have to log in through your mail ID and you can work. No need to carry the thing and go. Anywhere, any place, if you have a system, you can work. Okay. Then this is the link code.earthengine.google.com. If you go there, you can do the analysis, and this looks something like this. There are multiple things that here you can do. This main thing is here you can see the visualization of your maps, and top here you can write the JavaScript, or if someone is comfortable in Python, that also can be done in Google Earth Engine. Then the shape files, or we can say the location area file, that also you can add using this import feature here. Here in assets there is a new option from here, new files. If you have some files like that files you have created of your campus, that file you can import here as shape files. And the other files, if you want to download the boundary file of Dakshin Kannada or the boundary file of India, that also you can download from QGIS. In Survey of India also the boundary files are available, administrative boundary files of India. Okay, till village level it is available. Then using this code map.addLayer and map.centerObject, this is a JavaScript code. Using this you can add those maps to the map layer. You remember that map layer and code layer I showed you, no? It comes in horizontal bars. In the top you write the code, down the map visualization comes. Okay, so this is machine learning, this works in machine learning algorithm only. Like this you can add your study area map. Then the data that you want to work on, here I have worked on the Landsat data. And using the Landsat data, I have tried to identify what are the land features, land uses and land cover available in the Bangalore. You can do it for your campus and surrounding areas. Also, we can do it for the entire Dakshin Kannada district or your particular village. Okay, there is Landsat data is available for 30 meter by 30 meter. Then Sentinel data is there by 10 meter by 10 meter. So by 10 meter by 10 meter, I want to mean if the area is 10, how much is this room? 10 meter?
No.
Will it be 10 meter? Yes, no. Okay. So how many 10 meters are here in this room? Lengthwise?
50 meters.
Huh? 10 meter by 10 meter. How many will come in this room? If I draw a 10 meter by 10 meter box, how many of those boxes can I fill here in this room? 6 to 7. Okay. So so that means 6 to 7 pixels will cover this room in the Sentinel imaging. Are you getting my point? No. You got? Guys, you also got? Any confusion? So, in the ground, whatever the 10 meter by 10 meter area is represented, that 10 meter by 10 meter area will be represented by a singular pixel in the satellite imagery. That is what I mean by 10 meter by 10 meter data. In Landsat it is 30 meter by 30 meter. In MODIS it is 1 kilometer by 1 kilometer. Okay? Then you can see here I have created a false color composite. But this doesn't look like, we can't understand what this all colors means. Why it is all red? What these red colors and all those colors mean? This below thing we all know. This comes in Google Maps also. Correct? So, here from this map you can see, right? This is a urban area. All you can identify this urban area, right? No confusion in that? So, this urban area comes like this in false color composite. So what false color composite helps us do, here can you see this is my campus, this green patch. Can you all see this green patch? Can you all see how bright red it is? So the FCC tries to help us identify the vegetation area in red so that we can extract it better. The visualization of different heterogeneous land use features becomes easier in false color composite. That's why we visualize the satellite imagery in FCC. Okay, what is FCC? False color composite. Okay. So now we have seen how urban area looks in false color composite and how vegetation looks in a false color composite. Okay. Now this is a lake. Okay. This looks like this in a false color composite, dark blue or black. Then this is an open area. This looks like this light brown color in false color composite. Now you can understand how this false color composite helps us to identify different heterogeneous patches. Next is here I have done the land cover classification by classifying the area into vegetation and non-vegetation. How much percentage of the area is under vegetation and how much is non-vegetation? That here I have tried to compute using normalized difference vegetation index. That is called NDVI. What is this called? NDVI. Okay. So like NDVI, there are multiple indexes. Like NDWI, which calculates the water index. The NDBI is there, which is built-up index. So like that, there are multiple things. If you Google it, you will get it. It is like very easily available. So you can, using those indices, you can calculate the land cover of that area. Okay. Okay now. Okay. So here I have these green patches. Can you see these green patches? Those are the some green pockets in the city. All the blue represents the non-vegetated areas. That means the roads will be there, the open areas will be there, the buildings will be there. Okay. Then coming on to next, this is the land use analysis. In land use analysis, we try to see what are the land use featured in that region. So as I showed you the NRSC, based on NRSC color, red means buildings, blue means water bodies and green means the vegetation areas. So using this, we categorize the satellite imagery into different sections of land use components. Okay? So in your region also you can do it. Just you have to write the code and you can do it. This is the link where all the Google Earth Engine tutorials are there by Google Earth Engine people itself. You can go to that link and you can see the videos, and if you have any question, you can ask. Any questions? Can I ask you questions? Okay. What does blue color means? Water bodies, correct. What does yellow color means? Open or agriculture. What does red color means? Vegetation. Red color vegetation? Buildings. Are you sure? Okay. Then what will be the... If I want to map myself, I will map me as line, point or polygon? Point. Okay. If I want to map the whole classroom, then it will be polygon. Okay. What is the application that we will use in field to take the photo of the location? GPS map camera. What was the other app to take the point? what is the application to map those points? Google Earth Pro. What is the application to do all the analysis using machine learning? Google Earth Engine. Okay, thank you.
Okay, good morning to all. Now we will understand how soil is important to us and how to assess the quality of soil to for the survival of humankind as well as our Earth or universe. So, how is soil important for us? Can anyone answer? Why soil is important for us?
Agriculture.
Okay, crop production,
For vegetation.
for survival of vegetation,
Construction.
yeah, for construction purpose.
Degradation.
That is not a benefit.
Living organisms.
For habitat for organism. For carbon sequestration and mineral accumulation. So we know general thing that why soil is important for us. So we will explore how to assess the quality of these soils, and we will know the basic characteristics, physical as well as chemical properties of soil. So we can map. We shall assess the quality of soil in your neighborhood, where buildings are there, where forests are there, where vegetation is there. So all land use will have a different property, different physicochemical property in the soil. So we have 32 lakes in Moodbidre, and so you can opt for any one of them, and you can assess the soil quality for the catchment area in each lake. So one of the lake you can opt for that, and you can assess the quality for the different land use type. What is the texture? What is the quality of soil? What is the color of that soil? So all we will get to know. So this we have discussed why soil is important for us. So I will not talk so much. So soil is not simple a dust for us. It is more of that. It helps us to sequester carbon. It stores 3 times more carbon as compared to atmosphere. But our total atmosphere store carbon, it stores 3 times more carbon, and it helps in water filtration. Through what precipitation and rainfall we get, that water infiltrate through the soil, and it stores in the underlying layer, which gives us the pure water in the summer season. And it is a habitat for various organisms, microorganisms, and it provides habitat for all flora and fauna whichever exists in this earth. So this is the basic structure of soil profile. This is the vertical profile of soil. In the topmost layer consists of organic matter, which is very rich which have a rich mineral content. And below that, the topsoil have that similar but some compared to less fertility than this organic layer. Then we go deep into like B layer, C layer, and at the last we will get the hard rocks, means the big boulders in the underground layers. So there are various threats to our soil, like erosion, soil erosion, organic matter loss, the loss of topsoil, and nutrient imbalance. Because we grow food or crop for our sustenance, so we have a nature of greed in humans. So we think that if we will put more fertilizer, we will have a greater yield. But it is not a case. But instead of that, there is a certain limit by that a plant or that can withhold. So it can't take more nutrients from at a threshold limit. So all that excessive nutrients will accumulate in the soil, that will degrade the quality of soil, as well as with the runoffs or when the rains fall, it will flow to our nearby water bodies or storage. So it will lead to nutrients loading in that water bodies. That Gagana will tell you why that nutrients affect the water bodies. Means there will be a growth of microphytes and green cover that doesn't allow water to percolate through them. So this degrades the quality of soil as well as the water. Acidification, as we have as the time is passing, our soil is getting acidic in nature. That is not good for the crop production. So there is a major threat to our food security also because of that, and loss of biodiversity. With that, the main reason, the main cause is loss of biodiversity. We are losing all the microorganisms in the soil. And sealing means we are converting all these open spaces and all the open spaces into paved area. It doesn't allow water to percolate through the soil, and which is leading to runoffs, floods. So it is again the threat to human subsistence. So we should allow water to percolate through the ground, and we should restrict to build the paved surface. So now we will see what are the physical properties. As it is mentioned in the slide: texture, structure, color, density, moisture, permeability. So we will discuss it. So we have majorly three types of soil based on texture. Can anybody tell me what is the texture? What do you mean by texture? Not appearance. Means space. Anybody else? Yeah. That how we feel when we touch that surface. Either the surface is smooth or either the surface is rough. So based on that, if we touch the soil, we have soil, we have sandy soil. So it will have a rough texture. Means it will be like gritty kind of thing. So it will have a rough texture. As we compare to silty soil, it is less rough than as compared to soil. If we have clay soil, that is all sticky. We used to make idols from them. That is clay soil. That is sticky. So these are the three, and mixture of these three kind of soil, we will get. If we have a different combination, we will get the loamy soil. So sieve analysis can tell us the texture of soil, what we are trying to analyze. So by different size of this mesh sieve we have in our houses. So we have different kind of sieves to separate out the husk from our cereals what we cultivate from the finer cereal. So there are different kind of the sieve sizes or mesh sizes to analyze the size of the particles. So we will, for our analysis, we use a 2 mm of sieve size to analyze the texture of the soil. So how we will know what is the texture of the soil? So first we will take, suppose we are trying to assess the structure of soil from our garden. We will take the soil, we will collect it, we will put it in a jar, and we will put some water, so it will be fluidy. So we will shake it rigorously, and we will put it for some time so that the particles will settle down. So the particle which will settle first will be the gravels or the stones with the bigger pebbles. Then the sandy soil will settle after that. Then the silty soil. Then the clay soil. So clay soil will take a bit larger time, like compared to these two. It will take 8 to 10 hours or more than that to settle down because of the small particle size. And the topmost layer, what we in that this where the bubbles are there, that is a that matter of organic content where dry leaves and will be there. That is that organic content. So if we assess, we have this great this triangle pyramid. We can assess the quality of soil what we have collected based on, like in this soil, how we will get to know the proportion of soil different soil in that in that composition. So we will put a scale. We will measure the length of each type of soil, and then we will assess the what kind of soil is there. So in this, if we suppose we have 20% of clay, 20% of clay in that component, we will see the horizontal. You can see that clay, silt, and sand is a different dimension. So that clay, if we have 20% of clay soil, it will represent this in the horizontal direction. And if we have 55% of that composition is silty soil, then it will show there, and the sand. And if 25% of sand soil, then it will represent this intersection will represent the silty loamy soil. So this how you can assess the quality of means the structure or texture of the soil. So now coming to the soil color. There are different kind of soils with we can differentiate based on their color: brown, red, yellow, grey, black. Means this brown soil, what we see as usual in our gardens and other. This is the brown soil. Then red soil, where we use where the organic content is high. Means where you can see where groundnuts are. Means they used to produce groundnuts. There you can see that's iron oxide or red soil. Yellow soil indicates again the limited organic content. And black soil is the topmost soil where you can see near the lakes and where the means dense forest is there. You will see the top layer will be black is because of that degraded human leaves, and that is that black soil. And grey soil is where the water stagnation is there. You will see that grey soil because this grey soil represents the deficiency of dissolved oxygen. So you will see that, you will notice there. So how now coming to the density of that soil, how we will assess the density of that soil? We have a simple experiment. We have this instrument called core cutter, or you can take a PVC pipe, cylindrical pipe which have a fixed dimension. You can take that and you can collect a sample, a soil sample, and you can assess the density. You will weight that what we have collected in that sample, and you will weight it. And you will divide it with the volume of that core, what we have taken for the sampling. So you will get the field density. This is called the in situ or the field density, because we collect it. It contains various means it also contains the moisture also. So we will again if we require this will give the field density. We will then again. After coming to the lab, we will oven dry it, and we will take the dry soil. After removing the moisture content, we will have this dry soil which will give us this bulk density. What is bulk density? Bulk density is the density, means it represents how at what rate or how the soil will permit the water to percolate in the down layer. If the soil is clay, then it will have lesser bulk density, which means that it will allow lesser water to percolate through them, and the sand will have the higher permeability. So now coming to the permeability, as I told you that permeability means it allows the liquid to percolate through them. So this sandy soil will have the higher probability to precipitate to filter the water down, and this loamy soil will have lesser as compared to lesser ability to percolate the water than clay soil, because clay soil is highly rich in minerals, so and it is compact, so it will have a lesser ability to percolate the soil. Again, coming to the moisture content, how we will get the moisture of that soil? As we have assessed in during that oven drying, from that field density, we will assess we will separate out what is the bulk density. That remaining what we will get it the moisture content. Now coming to the these chemical properties of soil. Chemical properties means first we will see this soil pH. Soil pH, electrical conductivity, organic matter and other nutrient level. So soil pH will give us the alkalinity or the acidity of the soil, how acidic our soil is. So as the time is progressing, as I tell you that the acidity is increasing with the time. So we have a scale of 0 to 14 in which 7 represents the neutral and less than 7 represents the acidic nature of the soil or water. So greater than the 7 represent the basic nature of the alkaline nature of the soil. So we have this pH probe. Gagana will demonstrate you how to assess the pH or the alkalinity of soil using this pH probe. Now coming to the this electrical conductivity. Electrical conductivity denotes that how the our soil is conductive in nature, means what all minerals is there present in the soil, like we have salt, we have other metallic inorganic content, so it will show the conductivity of the soil. So this again we can assess this through this if there is a probe and we quantify it in microsiemens per centimeter. But there is no unit for pH, but it has a SI unit, microsiemens per centimeter. And similar to that, this total dissolved solid is we will measure that how all the organic and inorganic content in the soil, whatever the total inorganic, all the metals, all the minerals are there in the that soil sample where we are trying to assess. Organic matter is again the topmost soil. We will assess. There is a experiment to conduct it, but it is not represented here. But you can assess the organic content in the soil because of the microorganisms decomposition. Okay, thank you.
Okay, today I will be talking about monitoring of water bodies. So before I start, can you all, few of you, explain what are the importance of water bodies? Any few points? Any one of you?
Animals water, ನೀರು ಕುಡಿಯುತ್ತೆ. Filter, etc.
Mentioned here, it helps in maintaining and restoring the ecological balance, helps in groundwater recharge, and also for drinking purposes, and many more points that one of the person had explained. So those are all the important things the water bodies help us with. So what happens to these water bodies when different sources of pollutants enter? So the sources of pollution are from industries, our own houses, where the sewage is let or discharged into the water bodies, where these sources contain a lot of, as you can see, sewage and domestic waste, fertilizers from agricultural lands, pesticides, soap and detergents from our houses or the industries, and also heavy metals. So when these sources of pollutants enter the water body, and when the pollutant level is increased above the threshold level, what happens? They cause these three main or affect the water body. That is, the first one is increase of macrophyte growth. That is the aquatic plant. When the nutrient level in the water body increases, such as phosphates and nitrates, the macrophyte grow heavily. That is, you can see one of the lake has been completely grown with macrophytes. The second one is frothing. You would all have heard about frothing observed in Belandur and Vardur Lake, the catching of fire in those foaming that has taken place. So that is also because of increased amount of detergents that are being let into the water body, which contains mainly phosphates. Okay. The third one is algal blooms, where that is also due to increased amount of nutrient level. The algal bloom we can see the algal blooms. And also when the nutrient level is high, it leads to fish death, and also when the dissolved oxygen level decreases, it causes the death of fishes or the aquatic organisms that are there in the water bodies. So now we know what are the importance and what are the sources that are affecting the water body. What we can do is we can assess the status of water body. So the objective of water quality assessment is, first one, we will get to know whether the water body is polluted or not. And the second one is, what are the sources that are causing the pollution, like the site-specific sources, whether the water is being polluted at the inlet or the outlet or somewhere from the discharge of any industrial effluents that are being discharged at the inlet point. So those are all the things that we can assess. And the third point is, we can take up necessary actions after we know what is the situation of the water body from the first two assessments. Understood? Okay. So after we do the assessment, next we need to go and collect the water samples. So before collecting the samples, we'll be needing a few things. Before we go for the field visit, so these are all the things that we will be needing, which includes gloves, masks, 1 liter sample bottles, beakers, reagents, distilled water, BOD bottles, on-site parameter, probes and field notebook. So when you have all this, we will be going to a field visit. So when we visit a field, first you will take a walk around your water body, whatever the study area you have chosen, and you will analyze what is the biodiversity that is there, what kind of vegetation that is there around the water body. So the vegetation includes different tree covers that are there, you will note it down. And the third one is land use. Why we need to assess what kind of, note down what kind of land use that are there. See, if there is an agricultural area very close to the water body, when there is a precipitation happens, the rainwater runoff, the water that enters the water body through the agricultural area, carries a lot of fertilizers and pesticides. And if there is any urbanized area, the water won't get percolated, so all the rainwater runoff water from the urban areas, during the precipitation, what happens, it contains oil, grease and other dust particles from those areas that will enter the water body. That will also be one of the sources that will be affecting the water quality of the water body. And also you need to, if there is any waste dumping happening near the water body, you need to note down that also, because the leachate from those waste dumping will also affect the water quality. And also you will be noting down what is the water color, if it is too turbid, or as I showed you earlier, if there is any algal blooms which causes the water body to turn green due to excess of algae. And you will also note down what is the odor of the water body. So after noting down all these points, we will take the water samples at 3 main locations. The first one is at the inlet, where the water is being fed into the water body. And the second one is at the center, because it is not safe and accessible. You can take your water samples at the sides of the water body. And the third one is at the outlet, where the excess of water will be overflowing out of the water body. So these are not the only points that you need to take place. You can take as many samples as possible and do the analysis. So once you take the analysis, you need to write down what is your lake name on the water sample that you have taken, the lake name, the location, what is the sample that you have taken, the location of the sample point, the date and the time that you have taken. So after noting down all these points, we will be doing the water quality analysis. So there are two analyses that we will be doing. One is the on-site parameter analysis. That includes on-site parameter analysis, that is nothing but where we will be analyzing all these 6 parameters on the site where we have collected the sample. So you will be analyzing all these parameters at each sampling point. So those parameters include air temperature, water temperature, electrical conductivity, total dissolved solids, dissolved oxygen and pH. Okay? So the next is offsite parameters. These are the parameters that we will be analyzing in the lab. So these include biochemical oxygen demand, total hardness, calcium, chlorides, magnesium and alkalinity. So since you people can analyze even more parameters such as phosphates, nitrates and other heavy metal estimation can be done by you people in the lab. These are the basic 6 that I have written. So these are the standard methods that we follow. We follow APHE, American Public Health Association standard methods that are given by them. There are other methods also that you can use and estimate the water quality analysis. So coming to the first on-site parameter, the variation in temperature affects how the gases are being dissolved in the water, and also, for example, if the effluents are being discharged from an industry, if it is not brought down to the surrounding water temperature, like the regular average temperature, what happens, those effluents when they enter the water body, it causes a thermal shock, and also the variation in temperature affects the growth of juvenile fishes that are there. So to know what is the regular water temperature, we will be assessing the temperature of the water body, and also we will be noting down what is the air temperature. So to assess that, we will be using a thermometer. The analysis is easy. To know the air temperature, you will be holding your thermometer in the air. You are not supposed to hold it directly to the sun because it shoots up the temperature value. Make sure you are checking the air temperature in a little bit of shaded area. And also to estimate the water temperature, as you can see in the image, you will be inserting your thermometer in your water sample taken in a beaker or a Tarzan cup and estimate the temperature. This is the first parameter. So the second parameter is the estimation of pH. pH is nothing but we will be estimating if the water is acidic or basic. As Parasar already told, the range varies from 0 to 14. So the optimum range the water body should be is between 6.5 to 8.5. That is the pH where all the aquatic organisms thrive, like they grow well. As you can see, one of the image, they have shown the critical pH level that is required for few of the aquatic organisms. So to estimate that is the probe that we use, that is the pH meter. So this is the pH meter. So to estimate that, you will be taking your water sample in one of the Tarzan cups or a beaker. Take half of the beaker in half of the beaker full of water sample, and you will be immersing this probe in the sample and note down the reading. So as Sir told, there is no SI unit for pH. This is how you will do the pH analysis. And for the third one, this is total dissolved solids. So the sources for total dissolved solids are all the minerals, salts, organic matter and heavy metals from various sources such as industries or municipal sewage that are being discharged and various other sources. So what happens is when there is an increased amount of total dissolved solids, it reduces the water clarity. The water becomes too turbid. And also the second one is it increases the water temperature. The dissolved solids that are there, they hold up the temperature and increases the water temperature. And also the third thing is they decrease the photosynthetic activity as they don't allow the sunlight to pass through all the water body layers. This affects the photosynthetic activity. These are the few things that they affect. There are other things that gets varied if the TDS level increases in the water body. So to assess the TDS level in the water body, we have a similar probe. So this is the TDS meter. You can use this. So you take water sample in one of the beaker, immerse it, note down the reading. So this, the unit for TDS is ppm, parts per million. you will take down the reading in that, and also this probe gives your water temperature reading also. You will be noting down that also. And the fourth parameter is electrical conductivity. So it gives us the conductance of the water body. So the higher value of EC says that the inorganic impurities are high. So to do the analysis of this, as you can see in the image, the same probe similar to EC, we will be using it for TDS, we will be using it for electrical conductivity. We will be immersing the probe in water sample and noting down the points, noting down the data. The SI unit for this is microsiemens per centimeter. You will note down the value and test the next parameter. So the fifth parameter is dissolved oxygen. This is one of the very important parameter. So this is nothing but the amount of oxygen that is being dissolved in the water. So the main two sources for dissolved oxygen is, one is from the air, where the oxygen will get dissolved in the water, and the second is through the photosynthetic activity that takes place in the water body. So these 2 points are very important. So when we do the analysis, the first one is you will take a BOD bottle. This is a BOD bottle. So when you take the water sample, you need to take it in such a way that there is no air bubble formation in the BOD bottle. Because when there is an air bubble formation, it affects your DO value, it increases the DO value, because one of the sources for DO is air. So you make sure there are no air bubbles formed when you take the water sample. So after taking the sample, as you can see, you will be adding 1 ml of MnSO4 and 1 ml of alkyl iodide azide. And after you add that, you can see the brown flocculant that forms. This shows the presence of DO. If our water sample is too much polluted, the brown color will be completely white. The white flocculant will be formed. So after you add both of the reagents, you will keep your reagent bottle in a BOD bottle in a dark place for 10 minutes. So all the flocculants will settle down. Can you all see the third image? So after the flocculants is completely settled down, you will be adding 1 ml of sulfuric acid to dissolve the flocculants. The fourth image shows that. So from the fourth image, whatever the sample is there in the BOD bottle, you will take 25 ml of sample, add 2 to 3 drops of starch and do the titration against sodium thiosulfide. So your end point will be from blue to colorless. So you will note down your reading and inculcate in the formula that is shown here. So you can see one of my colleagues is doing the on-site DO estimation. You will be carrying all your reagents in those Tarzan cups and you will be estimating it on-site. So coming to the next parameter, that is biochemical oxygen demand. So water has microorganisms and they take up a lot of oxygen to decompose the organic matter that is there. So to estimate what is the amount of oxygen they are consuming to decompose the organic matter, we will do the BOD estimation. So this is what you do is you do the estimation of DO on the first day. On the same day, you will collect the water sample in a BOD bottle and keep it in an incubator for 5 days. And on the 5th day, you will do the BOD estimation and inculcate the values in the formula given below. So coming to the total hardness, so water, sometimes when we use detergents to get that lather or the foaming thing, we need to use a lot of detergents. That is because the water will be too hard. So the main cause of that hardness is because of calcium and magnesium ions. So to estimate the total hardness, we will be doing here. So to do this, first I am taking 25 ml of sample and I will be adding 1 to 2 ml of buffer solution. as an indicator, I am adding a pinch of heliochrome black tea. Now that gives me a pink color. Next I am titrating it against standard DDTA. So my final end point should be from pink to blue. So as you can see, this is my end point. So I will be noting down the end point, taking the value and the point, I will be inculcating that in the formula that I had shown earlier and estimate the total hardness. So total hardness, as I told earlier, is mainly higherly because of calcium ions. So to estimate calcium ion, I will do the similar titration, and for that I will be taking 25 ml of sample and I will be adding 1 ml of 8% NaOH and a pinch of mur oxide indicator. Then when I do the titration, my end point should be from pink to purple. So I will note down the reading and do the calcium ion estimation. So the next is magnesium ions. So to estimate this, there is a formula that is shown here. I will take the calcium ion, whatever the value that I have got, and subtract it with the total hardness value. So that gives me magnesium ions. Usually when we do the analysis, calcium ion concentration will be higher. But for our samples, when we do the analysis, if our magnesium ion concentration is higher, that is because of the presence of high algae in our water sample. So the next parameter is chlorides. So as we all know, water does have some amount of salt content present in it. So to estimate each of those ions, we will be assessing it individually. So to do the chloride ion estimation, I will be taking 25 ml of water sample and I will be adding 2 to 3 drops of potassium chromate and do the titration against silver nitrate. So that gives me the end point will be from yellow to red. You can see the formula that is shown below. You will be inculcating your value in those formulas and estimating the chloride ion concentration in the water sample. So water bodies, as we all know, they have a certain pH. Those water bodies, what they do is they try to keep their water in the neutral condition. Like they try not to have the water, get into that acidic condition. So to estimate this buffer capacity of the water, we will estimate the alkalinity of the water. So the similar way we will be doing the estimation. For this we use phenolphthalein and methyl orange indicator and note down the endpoints and inculcate the values in the formula given below and estimate the alkalinity. So after we do the analysis, whatever the data that we have got, we will be comparing that data with the CPCB, that is Central Pollution Control Board standards that are there. So you can see there are various classes that they have shown. The class A is for drinking water, the class B is for outdoor bathing, C is for drinking water after conventional treatment, D is for fish culture and other wildlife propagation, class E is for irrigation and industrial cooling. So when you compare this table with the data that you have got, you can say what is your water body fit for various. When you compare it with these parameters, you can say whatever the water sample that you have analyzed falls under which category. Other than that, you can also compare it with the water quality, drinking water quality standards that are there. You can analyze your AquaGuard waters and say or make sure whether they fall in the given required standard that are given by Bureau of Indian Standards. Other than this, so there are computer science engineers that are there. So we have a lot of water quality index equations that are there. It is like giving the ratings for our whatever the food that we deliver. We give a 5-star ratings, 5, 4, 3, 2, 1. So those water quality index, what they do is they give us the range and say whether our water quality is good, bad or excellent. So what you can do is you can develop a model where when we give so much of data that we collect after assessing the water quality, when we give those data in that model, it gives us the range after calculating all the weighted, all the unit weights, and after that as the calculation, we'll be getting the final output. So we can take up that as a project and do that also. So this is, Sir has already shown, this is our website. So Lake Symposium 2024, when you go to that, when you click on the environmental monitoring section, you will be taken up to this page, and in that section 7 gives us the complete detail on the water quality assessment. So thank you.
I'll discuss about topic biomonitoring of lakes. So biomonitoring of lakes means it's a monitoring of a water body with the help of all the biotic organisms living in water. Okay. So water gets polluted due to domestic sewage, then industrial effluents and agricultural activities. Through that, pesticides and insecticides can come into. So when water gets polluted, all the entire food web will be affected, like the primary producer, then the primary consumer, secondary consumer, tertiary consumer. So all the population of all these aquatic organisms will come down when water is polluted. So there will be complete decline in the biodiversity if water is polluted. So biomonitoring of lakes, mainly we will look at the microalgae or phytoplankton, then zooplankton and macrophyte. Okay. So phytoplankton, they are plant-like organisms. They have the chlorophyll content. They can have the ability to photosynthesize. So they are the phytoplankton. They move freely in water. So they are also called microalgae. So they are typically green in color. Next is zooplankton. They are animal-like organisms which freely moves in water with the help of water currents. So they are termed zooplanktons. Then we have macrophyte or aquatic plants. So you would have seen aquatic plants, no, in all the lakes here. Have you seen? So how do you collect phytoplankton and zooplankton? Let's see. So here we need a plankton net to collect this phytoplankton and zooplankton. So according to Ramachandra et al. 2008, if we pour about 50 liters of water through this plankton net, then all the algae will be collected at the tip of this. There's a small tube at the tip. So all that algae will be collected, algae and zooplankton will be collected in the tip. Then we have to transfer all the contents to another sterile container. Then go to the laboratory, look under the microscope. So this is how an algae looks like under the microscope. Okay, and these are the zooplanktons. Okay. So mainly in Bangalore lakes we have like the microalgae that. So in Bangalore lakes we have mainly 4 types of algae, that is Basidiophyta or also called diatoms, then Chlorophyta, that is green algae, then Cyanophyta, that is blue-green algae, and we have Euglenophyta. So all these algae differ according to their pigment content in them, their cell wall structure, everything is different. So according to the identification keys, we have to identify which algae or zooplankton is that. Okay. So only for phytoplankton and the zooplankton we need a microscope. All the other aquatic components like macrophytes, then fish, birds, we can use a camera for capturing them. Then zooplankton we have, these are zooplanktons. So they are animal-like organisms. So they are the primary consumers in an aquatic ecosystem. Whereas phytoplankton is the primary producer. Okay. So there are main 4 groups of zooplankton. That is protozoa. You would have seen like examples of amoeba and paramecium that you would have seen in the small classes, isn't it? You have read about it now. So the next is the Cladocera, which is a higher organism, and for example is Daphnia. This is Daphnia species. Hope you can see. Then next is this Copepoda. Copepoda, they have antenna which filters all the food in the water. So the next is Brachyona species which comes under Rotifera. So there also like microalgae, they have different characteristics and we have to identify it. Next we have aquatic plants in water. So they can identify according to the leaves, stem, roots, rhizome, etc. And there are also different types are also there. So there are 4 types, like submerged plants. Submerged plants, the roots they will be rooted in the water that is inside the lake, and then they will be completely immersed in water. For example, is hydrilla. This is hydrilla and this is ceratophyllum. Okay, so they will be completely immersed in water. Then next is free floating plants. So these are the plants which freely float in water. So their roots are not rooted inside the soil. So their roots will be in the water and the leaves will be up above the water. So they will be freely floating in water. But example is Iconia species, then Lemna, then Pistia. Then rooted floating plants. These are plants which will be completely rooted in the water, but their leaves and flowers will be freely floating in water. Some you would have seen lotus and water lily. They are all come under rooted floating plants. Next you have emergent plants, means they are the marginal plants or shoreline plants. So they will be completely seen near the shoreline. For example, like Typha species and then Colocasia, etc. Okay. So you can be thinking how we can identify all these aquatic organisms. We have lot of databases, like we have algae base for identify algae. Then Copepoda is for zooplankton. Then Plants of the World Online and Flower Database is for macrophytes. Then Fish Base is for fish identification. Then for birds we have AV Base, then eBird, then Birds of India. So we can go and check. So similarly we have, sir had already shown you BLISS, that's Bangalore Lake Information System, wherein we have documented 100 plus lakes in Bangalore, its water quality and all the biodiversity like algae, zooplankton and fish we have completed. Macrophyte and birds we have to upload. We are working on that. So the economic importance of algae. So which this is useful for your project also. So algae they have from algae we can extract biofuels like biogas, bioethanol, biodiesel and biobutanol. Then they also helps in the manufacturing of cosmetics. So they are widely used in the cosmetic industry because some species they have the antioxidant property, etc. So they are also used in the cosmetic industry. Then they have like some examples spirulina, which is having high proteins and other vitamins and everything. So they are used in the nutraceutical and pharmaceutical industry. They are also served as food. Then some algae have the ability for nitrogen have the ability to fix nitrogen from the atmosphere. So they are used as biofertilizers, and some serve as animal feed. So this zooplankton is widely used as live feed for the aquaculture. So likewise algae serve as food for zooplankton. So they are also serve as food. So what you all can do? One is like you can monitor the lakes in Mangalore. Okay, then document its biodiversity, and like us, you can also make a special decision support system which documents all the water quality and the biodiversity of the lakes in Mangalore. Okay. Another thing is you can check the ecosystem services like the lakes and biota. It serves all the ecosystem services like provisioning service, regulating service and culture service. That also you can account to. Then another is bioremediation concept. So the algae as well as the macrophyte, they have the ability to remove the contaminants from the water. They can remove, take up all the ions and nutrients, and then they will, as in the process, they will remove the contaminants in the water. So that capacity, that also you can check. So one project can be bioremediation capacity of macrophyte and microalgae. Next is biofuel concept. So I already told you algae can be used, the biofuels can be extracted from them. So you can extract any of the biofuel. So the long process you can do in your laboratory and then assess that. So another project is like I have already told you, like microalgae, they have the antioxidant capacity. So they are they are helpful for many like cosmetics and pharmaceutical industries, etc. So in your laboratory also you can just extract all those components which are useful and make it as a project. So these are some projects which you can do related to my project topic. Okay. So just want to say save lakes and biodiversity. Thank you.
Good morning everyone. Ma'am told us about the biomonitoring of lakes, meaning we will be seeing microalgae, zooplankton and macrophytes. Microalgae are the zooplankton and etc. Zooplanktons are the consumers, and macrophytes are plants like lily and lotus. Next come to plankton collection and identification. Their plankton net is used for the collection and the microscopic view is done to identify it as diatoms or zooplanktons or cyanophytes. Next, she told about mangrove lakes. We can see diatoms and cyanophytes and still more. Next are microphytes in lakes. There are 4 types. Submerged plants are, for example, it's hydrilla, and free-floating plants are pistia. And next comes rooted floating plants, that is lotus, emergentus colocasia. Next, database and identification. There are many websites as she has told, algae base and e-bird fish base, etc. And next is economic importance of microalgae. One is for cosmetics and biodiesel, biofuel, and it is also used as food. Spirulina, which has high nutrients, and animal feeds, meaning you can use it for feeding fishes, aquatic animals. The feed is zooplankton, and next is for fertilizers. Projects related to her topics are water-related monitoring of lakes, biofuel concepts, ecosystem service of lakes, and bioremediation concepts, and values added products extraction from microalgae. Thank you.
So you can extract that survey name, find out what is the biochemical composition and how you can convert that into value added product. Okay, that could be one this one. Whether you can convert that into bioplastic, then you can call that as Alvas plastic, market it. Okay, you can plan to do that. So I was planning to talk to Vivek to see that it is removed, but I thought it's better if students take up the project. When you start showing that there is a money with the lake, no, everyone will jump into the lake to study the lake. Okay, we will do that. How many want to work on lakes? Okay, reasonably good number. How many drink water? You all want water, but you don't want to monitor the lakes or water quality. Okay, now the next question is, we'll listen to Cincy about fish. How many of you eat fish? Okay, major section. Then how many like fish? Aquarium and all? Not many. Okay. So now she will tell you about the water quality linkages with the fish. See, if the water quality is, if a lake is polluted or the river is polluted, what will happen to fish? Huh? Fish also will have contaminants. You know, we did a study to find out what are the contaminants that are there in the fish. It has heavy metal. In Bangalore fish, we have heavy metal. You know, heavy metal like chromium, mercury, etc. are there in the fish. Is it good for to have heavy metal in the fish? So what will happen to fish eaters? Their kidney will fail, cancer will happen. That's what is happening in Bangalore. Now more and more people have lost in addition to brain, no, they have lost kidney also. So that is a serious thing. Now if you want to minimize that kind of problem in Mangalore and Mudvitra, how to monitor the water body to assess the water quality as well as the fish, and what kind of fish you get, you can also study. Okay. Now Cincy has come from Germany to take class along with Sulabha. So she will take a class and then she will go back to Germany. Okay. And for the lake symposium she will come.
Thank you, sir. Hi friends. I will tell about fish and water quality linkages. Okay. Before that, what is fish?
aquatic organism.
Yeah. What is the specialty of fish? No idea. Okay. I will tell you. Our fish is so papa. Okay. Without water, they cannot move from one place to another place. Okay. We can take elephant, we can take birds, or we can take any other organism. Some water crisis is there, or some other problems in their environment will happen. So they will move from one place to other place. Okay. But in our fish, our water is polluted. And first of all, where fish will land? Water. Okay. Then in our water body like river, streams, ponds, ocean, anything, the water is polluted. They cannot move from one place to other place. You understand the situation? We can move. Okay. We have any problem, we will move to one place to other place. Correct, na? We won't get proper food. Okay, wherever the food is available, there we will move and we will do our whatever work. That is not a problem. But this fish cannot move from, without water, they cannot move from one place to other place. So water is very important. Okay, only fish need water. We also require water. Then water is polluted. Only fish will affect? Huh? No. Okay. Near to your water body, like river or any pond or any lake, that water is polluted. You have any problem? You have any problem? Yes. Okay. Oh, you all are very good. Yeah. It is good. Yeah. I am very happy. Okay. Its water is polluted. That means our river water is polluted, lake water is polluted, or pond water is polluted. Then, we are not using the surface water. We are using always in the groundwater only. We are depending on the groundwater. But what is the problem? Near to your water, near to area, your water body is polluted. Then that place, that groundwater is also polluted. Then you will take that regularly, that groundwater for bathing or drinking and making food, everything. Then you will also, your body also contaminated and you will become sick. Okay, that's why you should, in your hand is our future. Correct, no? You all are engineers now? You please give the solution. You please give solution to your society. Okay. In this subject, what are the solutions you can give? You just find out. You can come up with the lake with that project. Are you interested? Yeah. Okay. This is our aquatic food chain. Here, in our aquatic ecosystem, we can see different types of algae. Okay, this is our macrophyte. This is our chota fish. Then these are zooplankton. What is she told now? That is Copepoda is this one. This is the rotifera. This is the cladocera. Okay, this is the debris. This all we can see in our water body. In our aquatic food chain, we have 3 different types of fishes like bendivorous fish, planktivorous fish and piscivorous fish. The bendivorous fish. First we can look bendivorous fish. Okay, in the bendivorous fish, they will take our detritus. That means degraded organic matters and all the things. Where we can see in the water of the particular lake or any water body. Then when they will hungry, you will go to cantina. That's why your fishes, when they will get hungry now, they will go to that water, the bottom of that place, and wherever that organic matter or something degraded matter, everything it is there, they will search and they will have. So you will make in the canteen, you will make the noise, na? Like that. Here also some disturbances happen, that is sediment. From that sediment, nutrient will enter into the water column. Okay, in that nutrients, our primary producer like microalgae or phytoplankton, they will take with the help of sunlight. Okay. The nutrients enter into the water body and the microalgae with the help of sunlight, they will make their own food and they will grow. Okay, you understand this situation? They will make their own food and they will grow. But in our zooplankton, they will go and eat this phytoplankton when they will hungry. Then another one is planktivorous fish. Planktivorous fish, that name itself, it is there. They will eat planktons like phytoplankton and zooplankton. That's why that planktivorous fish is came. Then the piscivorous fish, they are the carnivores. They won't eat that like our microalgae or macrophytes or something like that. They will eat our small planktivorous fish or fish. That's why that piscivorous. Pisci means fish. Fish eating fish, we can call it as a piscivorous fish. Okay. So they are very good, rich in protein and they are nutritious. That's why we also taking this fish. Okay. They are eating the different varieties of food items. That's why they are become the nutrient rich fellows. So we are eating that fish. We can see that because of that we will get the vitamin D, then treat depression, then protecting from age related problems, then reducing the risk of heart attack. Then it contains omega-3. Omega-3, that means eicosapentaenoic acid and docosahexaenoic acid. It provides the treatment for the cancer, asthma, cardiovascular disease, all the things. Okay. Another one, the fish growth. For fish growth, the biotic and the abiotic factors will affect the fish growth. What are the biotic factors? Here we can see that the prey-predator. The prey-predator relationship is very important. Wherever the prey is higher, then it will also affect the fish population. Then wherever the predator is higher, then also will affect the fish population. So the prey-predator relationship is very important. Another one is algae. That algae is good. They are eating. They are taking food. That fish will take algae. But what happened? The nutrient enrichment. You know that word, nutrient enrichment? Yeah. Why that nutrient enrichment happened? Because of agricultural activity, industrial wastewater directly entering to the lake, or domestic wastewater directly entering to the lake, then there is the higher amount of nutrients like nitrogen and phosphorus. So their amount is higher, then this algae will grow. They will get plenty of food, that phosphorus and nitrogen. So they will grow very well. We cannot control. There your help is required. You are an engineers now. You can control the nutrients. You can find out what are the solution to control that nutrients part. So I don't know who all are that here the civil engineers. Huh? Oh, everyone. Okay. Then all together can do the thing. Okay, then microorganisms, other or species like water birds, all the things will affect the fish population. Then another one is abiotic factors like temperature, dissolved oxygen, TSS, total suspended solids, then pH, light, mineral space, toxic minerals, then nutrients, etc. This all will affect the fish diversity or fish population. Another important thing, water is polluted. We can see here the fish will face the gill, jaw, eye, muscle, skeleton and fin deformity. Okay. Another important part is the split fins. The fish has different types of fins. Dorsal fins, pectoral fins, pelvic fins, then anal fin and caudal fin. These all the fins they are using for the movements and the swimming purpose. Okay, in that some splitting or something it happened, they cannot move properly, then they will die very fastly. Okay. Then why our fish is telling? Why our fish is telling stop water pollution? Okay, I'm not waiting for your answer because we wanted to stop this one. So stop. They are telling stop water pollution. Why? Because they are healthy, then we will also healthy because we are consuming that fish now. Then only we will get good health. They are not healthy, that means they will die. Then that water you will use for the other purpose, you will also get the health issues. That's why they are telling, they are given the priority to us also. That's why they are telling, please stop the water pollution. Okay, then water pollution because of different activities like agriculture activities, industries, fertilizers and pesticides, waste disposal, mining, then fossil fuel combustion and etc. So in water body, different types of heavy metals and pesticides, everything it will be entered. Then we will consume that fish or that aquatic products or that water, then we will get different types of diseases. Okay. Then another important thing is microplastics. We will use the plastic products, plastic bottles, plastic bags, etc. So from this, what we will, after our usage, we will throw. At last, it will come to our water body or some other places. Okay. Then, because plastics, because of sunlight and time duration, they will become split. Okay, they are different parts. That their size, the microplastic size is just 5 mm. Okay, in that one entering to the water body, then our zooplankton and phytoplankton will consume. Then small fish will take, then from that large fish, then that large fish will the human will consume that large fish. Okay, I am not taking fish, then why should I bother about that? You also bothered about these things. Why? Because you all are taking the bottled water, packed food items, everything. Then also you will get the microplastic in your body, the microplastic will enter, then it will affect your health. At least you please concern about your health and you should protect your water body, your environment. Okay now? Yes. Yeah. Then for fish study you can do these are some links, the India Biodiversity Portal, Animalia, then fish base, then IISC, that one, this is our group, we are made this based on the fish status and all. then the IUCN Red List. Why? Because we are finding all the fish species. In that fish species, what is the status? That is near threatened or vulnerable. That is the data deficient, least concern. That way you can identify the features. Then for fish study, you all are engineers. So fish diversity in particular lakes, then heavy metal analysis of water, then heavy metal analysis in fish, then method to remove heavy metal from lake water. Okay, then another one, microplastics in water, then microplastics in fish and its harmful effect. This all the project. Actually, we are given the question paper, we are given the answer paper. Just you come for the presentation, that's the thing. We given every idea, everything. That's so. Save lake for fish, do not pollute water body. Thank you.
Okay, who wants to summarize in 2 seconds?
She spoke about fish and water quality linkages, aquatic food chain, importance of fish. It is rich in vitamin D, helps treat depression, protecting from reducing age problems, reduce the risk of heart attack. Next is about fish growth. Next is about abiotic factors such as ammonia, nitrate, toxic minerals, space minerals like pH and etc. Next she spoke about morphological deformities in fish due to pollution, that is gill deformity, jaw deformity, eye deformity and etc. Next she gave some links about India Biodiversity Portal, Fish Base, Animalia Encyclopedia, ISDSS, IUCN Read List, and at last we have fish study, fish diversity in lakes, heavy metal analysis of water, heavy metal analysis in fish, methods to remove heavy metals, save lakes for fish. Thank you.
Where the lake symposium details are available, you can see that wgbis.ces.iisc.ac.in, wherein you can go to the lake symposium. What Vinay? Material that I think Sulabha, Sinsi and Gagana showed. So you can look at those reference material wherein you get the details how to do the experiment, etc. Now we have said why you have to do the experiment. What is the purpose of that? See, in this case you get the reference, there is a student and teacher corner wherein you get the videos also. There are videos related to birds, butterfly and water quality, etc. Then this reference material will give you the various documents. Suppose if you want to do my village biodiversity, there are students from science background. You can do that. You are in your village, what kind of diversity of plants and animals are there, you can document that, okay? Then water quality assessment, etc., you can do that, okay? Then there is a format for documenting the biodiversity in this link. Then there are some reference material videos are available. Then we have conducted this pre-lake conference at various institutions. We have put the materials, what we have used, the videos are available. So you can refer that. For example, if you look at the Purna Pramati, this one, you will see the presentation PPTs are available as well as the videos. You can refer that. So like some of you, the computer also has become tired. So I will give a rest to the computer. So I wish you all the success. I hope you will do a good job and all of you will take part in the event. Next time, I want each one of you to attend the symposium and then take active part. See, now the program is like this. Today we had orientation program. You will do the projects. And end of July, your teachers will evaluate and send the list to me. Okay? And of course, when your teachers evaluate and shortlist, they would have given some suggestion to you all. So you can implement those suggestions. In August, I will come, my team will come, we will do the evaluation. And out of those selected by the teacher, suppose there are about 200 youngsters here. Let us say 100 projects will be there. Out of 100 projects, teachers might shortlist 40. Then that project, 40 project, we will come and evaluate. If all 40 are good, we will select all 40 for the final round. If there are quality reasons, then we will screen that, okay? That is our idea. The team can be single person or 2 person, that is good, but not more than 3, the team, okay? You can take up the work related to the both science and engineering. Don't think that I am a civil engineer, I cannot do anything. Our Vinay has demonstrated what a civil engineer can work on, the various aspects. So similarly, biology or science students can do the project related to science. Engineering students can integrate science and engineering and come out with an interesting thing. See, I am sure all of you have brain, no? I am asking a question. Yes. You know, I want you to raise hand to tell me how many of you have brains. Okay, that is good sign. 1 or 2 didn't raise hand. I don't know why it is. Okay, I'll accept that. Now, you know you all have brain. Now you prove that you have brain by taking up a challenging work, okay, innovative project. Come with a challenging work, okay, so that we should also feel that you have done a good job. You know, teacher gets satisfied when student does the excellent work. We look forward for that. See, this event happens once in 2 years. You are lucky to be here because of Vivek Alvas. Had he not come to my office asking me to organize the event, this would have been in one of the colleges in Bangalore or at Indian Society. So now we have come to your institution. We are your Adhitis, your guests. And you need to learn how to treat your guests. My black shirt man, next time when I come, don't talk in the classroom with your friend. Don't think that you are the only intelligent fellow. Vinay has said that by 12:35 or 40 I should wind the session. So now it is 12:40. So I am handing over the mic to Vinay. Okay.
Okay. Just to put across few words, probably we are from different departments: civil, mechanical, electronics, computer science. We have people from MSc. We have people from agriculture. So the idea was to give you a glimpse of what you people can do. So as an individual, as a particular department per se, you will not have a clear-cut idea. Probably if I talk about computer science people, they take data from a different source, something like Kaggle or something like other web pages. Rather than taking some from web pages, you guys can connect with other departments wherein they can generate data for you. So you guys can work together. Probably this will be a baseline for you where you can go with interdisciplinary research. So having said this, I request Akash to give a vote of thanks.
ಬಗ್ಗೆ ಮಾಹಿತಿ ಕೊಟ್ಟಂತಹ ಡಾಕ್ಟರ್ TVRAMACHANDRA, ತುಲಿಕಾ ಮಂಡಲ್, ಪರಾಸ್ನೇಹಿ ಗಗನ, ಸಿನ್ಸಿ ವರ್ಗೀಸ್ ಮತ್ತು ಅಸುಲಭ KS ಈ ಎಲ್ಲ ತಜ್ಞರಿಗೆ ಆಲ್ವಾಸ್ ಶಿಕ್ಷಣ ಪ್ರತಿಷ್ಠಾನದ ವತಿಯಿಂದ ಧನ್ಯವಾದಗಳನ್ನು ಸಲ್ಲಿಸುತ್ತೇನೆ. ಹಸಿರೇ ಉಸಿರು, ಕಾಡು ಬೆಳೆಸಿ ನಾಡು ಉಳಿಸಿ, ಪರಿಸರವೇ ಗುರುವು, ಪ್ರಪಂಚವೇ ನಮ್ಮ ಕುಟುಂಬ, ವಸುದೈವ ಕುಟುಂಬಕಂ ಎಂಬ ಅನೇಕ ಸೂಕ್ತಿಗಳನ್ನು ನಾವು ಆಲಿಸುತ್ತಾ ಬಂದಿದ್ದೇವೆ. ಆದರೆ ಅದು ಬರೀ ವಿಚಾರಣೆಯಲ್ಲಿ ಅಲ್ಲದೆ ಆಚರಣೆಯಲ್ಲೂ ಇರಬೇಕೆಂದು ನಿಮ್ಮ ನುಡಿಗಳಿಂದ ನಾವು ತಿಳಿದುಕೊಂಡಿದ್ದೇವೆ. ಧನ್ಯವಾದಗಳು ಸರ್. ನಾವೆಲ್ಲರೂ ಸೇರಿ ಕೆರೆ ಸಮ್ಮೇಳನದಿಂದಲೇ ಆರಂಭಿಸುತ್ತಾ ಪರಿಸರ ಪ್ರೇಮಿಗಳಾಗಿ ಪರಿಸರ ಸಂರಕ್ಷಣೆ ನಮ್ಮೆಲ್ಲರ ಹೊಣೆ ಎಂದು ಮನಪೂರ್ವಕವಾಗಿ ಭಾಗವಹಿಸಿ ಯಶಸ್ವಿಗೊಳಿಸುತ್ತೇವೆ. ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಯಶಸ್ವಿಗೊಳಿಸಿದಂತಹ ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಧನ್ಯವಾದಗಳನ್ನು ಹೇಳುವುದರ ಮೂಲಕ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಕ್ತಾಯಿಸಿ ಪರಿಸರ ಹಾಗೂ ಪರಿಸರ ಪ್ರೇಮಿಯಾಗುವುದಕ್ಕೆ ಪ್ರಾರಂಭಿಸೋಣ ಎಂದು ಹೇಳಿ ನಿಮ್ಮಲ್ಲಿ ಮನವೊಲಿಸಿಕೊಳ್ಳುತ್ತೇನೆ. ಧನ್ಯವಾದಗಳು.